Hello and good evening. Well, this is evening here in or nighttime in upstate New York, and welcome to Connected Conversations. And I am thrilled to have with us today to talk about soil microbes, Dr. Priyanka Kushwaha, and she is a postdoctoral research associate in the Department of Environmental Science at the University of Arizona, Tucson, and received her PhD in soil microbiology from the Florida International University in Florida, and studies the kinds of soil microbes that are present in nutrient limited, arid, and metal contaminated soils, and what role they play in providing nutrients to the plants. So welcome, Priyanka. I think that was a good enough intro. I think I covered
Yes,
everything.
it was. Yes, thank
Awesome.
you, Scott,
Glad to
for
hear
the
that.
introduction. Yeah.
Glad to hear that. I always
Yeah,
worry.
that broad overview of what I do.
Oh, all right. We already have Phillips here. Welcome in. And if you got any questions, don't forget to ask those in chat. Glad to see you here tonight. So tell us first about, I want to know, like, soil microbes. What got you interested at? Did you like drop some food on the floor and you're like on the ground at a camp and you're like, oh, I really should I eat this or not? Or was it
I
some
wa-
other formative
I
moment?
wish it was like that. No. So I actually stumbled upon soil microbiology from wanting to do forensic science. So it's like a completely 360 turnaround for me. When I came to US for my PhD, I'm originally from India. I was looking for PhD research in forensic science because I was very fascinated by the application of science, using it for forensic science and provenance purposes. And I learned about a project in my advisor's lab at Florida International University where you could use soil microbes and profile them from different soils, and you can identify if the soil is coming from a particular region. So you can compare two soil
Right.
samples, and if they have similar microbial profiles, and what I mean by microbial profiles is similar to having human profiles, like our DNA fingerprinting. So we can identify us from our parents or siblings. So markers like that you can use. So I was fascinated by that. But when I started exploring that more, I was like, oh, wait a minute, there's more to soil microbes than just using them for forensic science because they do a lot more functions in the soils. And that's how I made that leap from forensic science to studying more microbial functions for plant purposes.
Awesome. That's kind of see that transition now, because I was always fascinated by that too. And like, even like in Sherlock Holmes, it'd be like, ah, the mud on your boot tells me that you were down by the Thames,
Yeah,
and
so most, yeah, definitely. So most of the analysis on soils have been more chemistry based, like matching the chemical
Mhm.
profiles of these soils. But in the last 10, 15 years, we are also looking at DNA markers, and that's how soil microbes, even plant DNA has been used. So there are
Mm.
different, other than the chemistry, there's more biological aspects that are being used for comparisons. Yeah.
It makes sense when you think about it.
Yep.
Yeah. Um, and so I guess that makes a good transition, but so you were kind of like, I really don't like forensics so much. I just like the microbes themselves. Or
Yes, part of it, yes. I didn't say that I didn't like forensic anymore, but I was like, oh, microbes, they do a lot more, and there's more like if I were to focus on forensic science, it would be very narrow of a scope, but studying soil microbes just allows different aspects of the research. I can look at it in an environmental question, and I can look at it in an agriculture setting. So it just opens up more ways of understanding what microbes are doing in the soils.
it, I'll wait till Philip gets back till he answers his question. Unless he's back. Tell us if you're back, Philip. So what exactly are microbes doing in the soil? They actually do things?
Yes, they do. Yes. It's interesting. It's fascinating that I didn't even like pay attention to soil microbes ever, like when I was in school, because nobody really emphasized that on me. But soil microbes are doing a whole lot in the soils. So just for starters, that's the nutrients that are available in soil. They are there because of one of the application of microbes, because they are changing it from one form to the other, so it becomes more available for the plants to uptake. And be it carbon, nitrogen, or phosphorus, there are all different kinds of bacteria that are playing a role, and even fungi for breaking down complex carbon in the soil so it can be available for the plants to use. And I'm talking, the functions remain pretty much similar, but when I'm referring to nutrient cycling, I'm more thinking about ecosystem services. So, in
Hmm.
terms of more natural ecosystems versus when we are talking about agriculture or even gardening in our yard. It's similar because there are still microbes in the soils and they will do similar processes. But in agriculture, we are adding chemical fertilizers. In our gardens, we are adding potting soil. So we are manipulating some of the nutrients by adding chemicals in our soils too.
All right, everyone get all that? Um, so to the question, are there good and bad?
Yes, there are good and bad. So, another thing to keep in mind is microbes are not bad, like inherently bad, like they're not bad microbes. It's just a matter of opportunity. So, microbes in the soils, they're trying to get nutrients for themselves, and in the process, they help the plants. But sometimes, because there is a disbalance that
Hmm.
occurs, that can make them opportunist, and in that scenario they can cause plant diseases. So mostly they're good, but there are some which are bad.
Okay. And I guess the question here isn't the weed killing chemicals in the soil doing bad things? Like, do when we add herbicides or pesticides, does that affect the survive? I guess, do that affect the microbes?
There have been, obviously I'm not looking into that, so my
Yeah.
research is not focused on that, but definitely any kind of chemicals or even bio more greener chemicals that we use to kill the pesticides, they have some sort of effects on the microbes. And I
Hmm.
think there is research to support that if it's changing the microbes, then is it gonna be harmful in terms of the nutrient cycling for the microbes
Hmm.
to help the plants.
Now I'm thinking back to high school or even like middle school
Hmm.
biology for anyone that's taken those courses, which is almost everybody. And they have this the decomposers, right? You have all your different things. You have your herbivores, carnivores, omnivores, decomposers. So this is what we're talking about. We talk about these soil microbes. Leaves and other things fall to the ground, and your decomposers, like the bacteria, break it down, right?
Yes. Yeah, so when we're talking about like leaf fall, so that would be litter, and there are specific group of bacteria which are trying to break down those carbohydrate molecules in these plant matter, and then they help with the release of making those complex carbohydrates into simpler forms, and also that helps in the process of creating that organic matter that we talk about in soils of being a metric for well, a good metric for having plant support. So most of the temperate and tropical soils, they will have more organic matter. So you see the dark black, brown color because they have that higher organic matter, versus when I'm in a desert, I don't see that color. The soils I see in Tucson, they are more orange because of the iron in them, or they're more on the reddish brownish scale. So
Mm-hmm.
they have very less organic matter, so low in carbon in general.
All right, so it gets into what Eagle Scout Survival Tips is talking about. Your soil is made up of rocks and stuff, but there's also organic matter in there, including those microbes.
Yes, yes, that is correct. Yeah, so soil is formed from the weathering of rocks, but at the same time, because of the vegetation and all the plant material that gets accumulated, that will, and with the microbes, it will result in organic matter creation.
Now, this begs the question what you do research in, though. When there's
Mhm,
not a lot of organic matter falling down into or onto the ground,
mhm.
there's not a lot of food for the microbes, but they
That
still
is
survive somehow.
Yes, so they have adaptation strategies. So microbes are pretty good because they are, especially bacteria, because they are unicellular. So they don't have a lot of complex machinery as we humans or any other higher forms of animals have. So what they do is, and which is seen in the category which is the most predominant in desert soils, which are the actinobacteria. The certain groups in actinobacteria phylum, what they do is they make spores. So when it's extremely hot and there's less water availability, they will make spores, so they don't have to spend energy in doing any
Mhm.
of the cellular processes, and as a result they don't need as much nutrient during that time. And then whenever we have a monsoon, which has been rare, but this year was good, they will get activated because now there's flow of nutrients in the soil, and then the spores will go back to reproducing and having that cellular mechanisms activated.
So if we look at some what seems to be like a chemically caustic extremophile condition, is there still a lot of biodiversity in the microbes? Like if I pick up a spoonful, is it just chock full of like hundreds of different species?
There is. They might not be exactly comparable to more rich soil, but there is still diversity because the selection has happened for microbes which can live under these extreme conditions.
Hmm. Now, this also makes me think on Mars, right? There's soil in a
Mm-hmm.
way.
Yeah.
Could there be some spores in there that occasionally reproduce when it's just the right conditions and then turn back into spores again?
There's definitely a possibility. And there is not the work, well, not something that I worked on, but there has been an article that was written by one of my bosses, and she sort of compared the extreme conditions in a desert. She worked in the Atacama Desert in Chile, and she compared the
Mhm.
conditions there because it's so dry and hot to what the conditions are in Mars, and there might be some microbes which are present in Mars, which are also found in Atacama. I don't know if that research has been really done or published, but there is a possibility. There's a lot more to explore in Mars currently.
Oh, there is a lot more to
Yeah.
explore, but
Yeah.
that's also why they decontaminate those rovers so much so they don't bring those bacteria because they could
Yeah.
potentially survive the trip,
Yep.
there for
Yeah.
sure.
Yeah.
Um,
They're used to being in extreme conditions, and it only takes like one non-native
mhm.
bacteria to make the trip.
Yep. So Phillips has some more great questions. We can kind of veer from the extremophile and go to that question. Uh, does the microbes help the plants grow? That's a great question.
Yes, they do. So going back to what I said, that they are recycling the nutrients. So plants have to have nutrients available to them in a form that the transport channels that plants have to uptake those nutrients. And sometimes those nutrients are not available for the plants to uptake. So what soil microbes are doing is that they break it down into a more simpler form for the plants to. So indirectly, that's one way how the bacteria will help. Fungi play a different role. What fungi do is they create these structures around the roots, which are called hyphae. So it's more of like a symbiotic relationship where fungi is getting some nutrients for the plants and then helping the plants to get nutrients. So fungi spread all across the soil. Um, there's a limitation to how far they can go, but they still can scavenge out to get those nutrients for the plants. So in that manner, they are bacteria or fungi, they're helping get those nutrients to the plants. And they're also some bacteria which are called nitrogen fixing bacteria. They also run a symbiotic relationship with plants, and they are more in leguminous plants, where again the bacteria gets the carbon from the plant, and then it provides that nitrogen that the plant needs for its mechanisms and growth.
Funguses are decomposers?
Yes, they
Yes,
are.
they are.
Yeah,
Very good.
yeah,
A plus.
yeah. They do break down organic matter. I'm sorry, complex. Yeah, so they make decompose the carbohydrates and
Mhm.
also help with breaking down the organic matter into simpler forms.
Now I find it interesting that like the bacteria have a symbiotic relationship with the fungus, and the fungus has a symbiotic relationship with the plant. And some plants also have a symbiotic relationship with the bacteria. It's just a one big happy family. Uh,
For most parts.
you know, and it's interesting. When I also think that we as humans have the bacteria on our insides, plants have their bacteria on the outsides. you know,
No, they have some
they have
insights
inside
too.
too, but it's like,
Yeah,
but as far as that carbohydrate breakdown,
yeah,
they're
that
breaking that down
happens.
outside, and then so I don't
Yeah.
know. That's just me thinking.
No, no, no. That what you said is correct. That yeah, that is happening, and it's
Mhm,
like inside out of what we
mhm.
have inside versus plants have it outside.
And it's also everything is connected, and then
Yeah, yeah. Soils are pretty complex. There's a lot going on in there, biologically, chemically. So they are one of the, I personally feel, not just because I study soils, but I think soil is one of the complex mediums to study because of different aspects involved.
so you love it, but do you ever get like overwhelmed by it though? You're like doing an assay or something, and you're like, oh my gosh, there's just so much stuff in here. Or usually you're in such an extreme condition, you're like, it's not too bad.
No, not. I think it depends. So, most of my work is like extracting DNA and looking at like profiling the microbes who are
Hmm.
present. So that is not that hard. It's tedious extraction process, but it's not that hard. It was more of a challenging when I more of a challenge when I started my postdoc and I was trying to work with these nutrient limited soils and trying to measure enzyme activity by microbes. And then because everything has lower detection limit, it was just hard. I'm like, there's so many papers out there that have done it, but why can't I replicate it? But then I realized, okay, these soils are just not at the level at as other papers have published work in. So that was a challenge. But it's yeah, it's not overwhelming all the time. Some days are better than others. Yeah.
So do you do like the field research as well as that lab work? Like do you go out and get samples as well, or are you like a lab jockey? You're just in there.
No, we do go out in fields. Ours are our most of our studies, and it's just the nature in my lab that they are not temporal studies. So we're not going out regular like every year. There are other projects in the lab that I'm not involved with. They do it every year. So they have like spring set for going to different places. But I did do a couple of sample collections where I went out. And the first trip I had was in October when I had just started my postdoc, and it was still like 9,500 degrees, and there's no shade in the desert. So it was an interesting experience to go out and collect samples in the field.
That was my next question. Would be any fun field stories? Um, because scientists always have good ones usually.
Fun stories. Not that we almost no. I think my first experience was a little bit hard because I wasn't used to the desert at all, and then we were out in 95 Fahrenheit. But then we got used to it. I'm not even a morning person. When there are field trips, we know we have to start at 6 because
Mm.
if we can do most of our work in early mornings, it's just better for us because we don't want to be dehydrated when we are doing field work. And also, just to give a little bit perspective, it's a lot harder to collect samples in the desert versus in other systems because here you have to literally use a pickaxe and like dig holes. And in other climates, you can use a soil agar, which is a cylindrical tool that you get that you can like push it. It has a little metal bar on the top, and then you can just like push it into the soil. And because the soil is soft, you can collect it in that tube, which you can't really do in the desert because it's so compact that that soil auger wouldn't go in the soil at all. So we have to dig
Hmm.
holes, which is a little bit workout while we are in the field.
How deep of a hole do you dig?
We don't dig so most of our studies are between 10 to 20 centimeters, so it's not that
Okay.
bad.
All right.
Yeah,
That's not too
but
bad.
it's still, yeah. But if you're collecting a lot of samples in a day, then you are constantly digging, which
Okay.
is which is a little bit challenging.
So I dropped my candy bar in the desert.
Uh-oh.
How should I be worried about the bacteria I'm about to eat? Or I just brush it off and I'm good?
Mm, I would say no, because I think in general, I have been asked this question before, and people have different answers. It's not that I don't do it, like 5 second rules, I do have that, but I don't do it in the field because, especially in Arizona, we do have valley fever, and we don't want in any way to get exposed to that
Mm-hmm.
while we are on a field trip. So I would avoid doing that. Five
I
second
would
rule
avoid it
doesn't
too.
apply. I say, yeah.
I just wonder how many 5 second rule studies have we done?
Just at my house.
Oh, this is a good one. So when we plant something in the garden, it is advised that we need to let the soil rest for a while after we harvest the crop. I wonder what benefit this practice brings to the soil.
What are
Hmm.
they referring to rest as in like before replanting?
Yeah,
Can
I think so. If you want to elucidate and go in a little more detail, Michael, you can. Um, but there is something said for that, like the crop rotation and making sure there's the beneficial fungi
Yeah,
as well
so
as bacteria in there.
so again, that's not something that I'm 100% sure about because that's not what I work on. But I would, yeah, what you were referring to, Scott, as the crop rotation. So the idea of the crop rotation is to use crops that can help sort of restore the nutrient balance before another crop is planted. So depending on, I think it depends on what you are planting in your garden, and will that deplete a lot of nutrients from that
Hmm.
soil, and then you need to maybe add more. You can uh, re regrow something when you're adding potting soil or you're adding more nutrients to them.
Hmm.
So I think crop rotation sort of comes in where it was more popular when we weren't adding a lot of synthetic fertilizers in the field. So we were trying to minimize that and then using more like natural and green way of doing that.
Hmm.
That's my understanding of it.
Right. Right. And it's also kind of letting it go fallow, so you just let it stay for a while and replenish good nutrients and good bacteria.
Yeah, I think, yeah, it doesn't hurt to do that. Yeah.
Hmm.
But yeah, there's a limit to how long you want to let it be fallow for a
Okay.
while,
There's
I
a
guess.
question up that we did not answer
Hmm.
uh, from Mrs. Jinzo. Is mycorrhizal fungi the only fungi that partners with plant and tree roots? I don't know if you familiar with that or not.
Mycorrhizae definitely is the one that interacts the most. Actually, this in one of my studies, I did say like found some papers where I had to give reasons for why I was finding mycorrhizae in soils which don't have plants. So I did find some. I was like, yeah, that's interesting because mycorrhizae are supposed to be associated with plants. So most of the mycorrhizae do associate with plants, but there are some other groups too which might not particularly form hyphae with the plants.
Hmm. Well, you would think there'd have to be some mycorrhizae just there without plants, because what if a seed were to like flutter along and then land? You know.
Probably,
and
yeah, I don't
germinate,
know.
unless it was bringing some of the, I mean, it probably brings some of the bacteria with it and the fungi with it, but at the same time, it would grow better if there's already something just kind of waiting. Uh, I'm just,
Yeah, that's a good point. So one thing, there were two things that I sort of, so this was a comment by a reviewer when I was submitting my paper, and then I looked into the literature and I did find some examples, not many, but some examples where there were some free living mycorrhizae. But there's another hypothesis out there which is called the fungal loop hypothesis. And in
hmm,
that we talk about how this network grows between the network of fungi. It grows between plants, and that's how the nutrients are being transferred from one location to the other. So probably if I found, I hypothesized that it, because I found mycorrhizae in those gap areas, they are more of like a channeling
hmm.
fungi there instead of being associated with the plants.
I have heard that as well. And this is the whole idea. And I forgot who did that, the TED talk about the plants that are cooperating versus it being a competitive, forest, that
Yeah.
the plants and the microbes and the fungi are all helping each other be better versus this, the
Yeah,
idea
it's
of
this.
more Darwinian, competitive selection.
Yeah, it's mostly and I would like to believe, even if it's not true, that there is like a cooperation and support network that's in the soil, that everybody gets the nutrients they want and everybody is happy and healthy.
Mhm. I thank my bacteria all the time for helping and cooperating with me.
That's great. We all should do that because when there's a disbalance, your body doesn't like that.
No, you know.
Yeah.
Eh. Um, is there a particular like microbes that you find all the time that's extremely common, or is it always just like different? Like it just
No,
surprises you all the time.
no, so the way I study them is, and just to clarify, there's a lot of microbes that we study, they are still not culturable, which means that we cannot
Hmm.
isolate them and grow them individually. So, what we do is we use DNA sequences to look into which of the microorganisms are present, but at the same time, because most of them are unculturable, we don't get up to the species level classification for them. So we
Hmm.
have to look at the broader hierarchy of taxonomy, whether we are looking at phylum or order. And in all the soils that have been studied, there are certain groups, especially at the phylum level, that that in these kind of soils you will see. They can be changed a little bit in the different classifications that you find, but mostly, like
Hmm.
for example, I was mentioning in the beginning that actinobacteria are present in the desert, and they're present in the abundance of actinobacteria in the desert is more versus in other soil types, whether it's temperate, tropical, or grasslands. And in grasslands or in other systems, you will find
Hmm.
that acetobacteria and verrucomicrobia, they are more in abundance compared to what you see in the desert. So I think it's more of the adaptation to the kind of environment or the soils the microbes are, because as I mentioned, actinobacteria have bacteria which are tolerant to heat, UV stress, so
Hmm.
it's logical that they're found in the desert.
Makes sense. Um, yeah, I'm trying to get my head around this idea that there's so much that we don't know as far as down to like not even like the genus level, but like
Yeah.
only the. I mean, it's like that seems like wow, we are pretty ignorant. Um, yeah, in a way, you know. There's a lot more research to do, everybody.
Yes, it is, and we have to always remember that these there are certain theories about that isolation might not be applicable because these microorganisms, they only know how to live in a community. So to extract them out, because we have to realize that they're dependent on each other
Hmm.
for the nutrient or whatever they need. So when we try to isolate them, if we don't have the exact same environment that they had in natural systems, then it will be harder to isolate them and study them. So that's why it has been challenging to find the right kind of media to grow the bacteria. And that's why we say only
Hmm.
1% of all the bacteria are culturable.
Hmm. So we have some other great questions here. Uh, can soil microbes help to revitalize unfertile soil?
To some extent, yes, I would say yes. It might need more than just microbes. So it might be a combination of using some natural fertilizers, so adding some nutrients and then using microbes along with them to sort of help with the kickstart of that cycling that happens in the soil. So there is a possibility of doing that.
Mhm. And are there any bacteria that are damaging to the soil fungi?
Not that I know of, like for sure, but I'm sure there might be few which are like, no, I don't like this fungi, but mostly they are cooperative.
Yeah, I would think maybe if I always think of I think of something being damaging to like a community, it's usually an invasive of some kind.
Yeah, so yeah, you're correct. So in the microbial world too, if you have there are chances of more something damaging if it's not a native to that particular soil. But
Hmm.
sometimes I think it depends. Sometimes they may like the new bacteria that are being added to the soil, and that can help the plants. So
You would have to do an experiment.
yes, yes, do an experiment to figure that out, or look for it in the literature if it already has been done.
Right. Check the literature and then apply for your PhD. Yeah.
Yes.
um, so I was wondering, like, what, and we've kind of alluded to a lot of this in the conversation for your research. What benefit to like humanity do you want to help with? Like, what can we expect to maybe get out of this that might help us?
So we didn't talk about the other. I basically talked about one of my projects. I'm doing
Hmm.
another project too. But in the terms of studying soils in the desert specifically, we're trying to understand that when as we have noticed, we have extreme weather events, right? There are
Hmm.
fires in different parts of the country, so there's extreme heat everywhere. So as that continues to keep um, as the planet starts to get more and more hot, what is going to be the effect on the microbes? So, will the microbes be able to still survive and have those same functions and support the natural plants? Because, at least in my case, I'm not looking at a crop or agricultural plant, I'm looking at natural plants that grow in the desert. So, what will happen? Will the microbes be able to adapt? And their functions will remain the same and hence they will be able to support the plants, or if they can, then what will happen to the soil? That means if there are not enough microbes to support the plants, the natural vegetation may die off, and as a result we get desertified soil, so there's nothing growing in it because there's no nutrients. So I think that is the implication of looking at microbes in extreme temperatures right now. And the other project that I'm working on, where I'm looking at metal contaminated soils, is to understand um, inhalation. Not actually, I'm not looking at inhalation of dust particles for the neighboring community from where these metal contaminated soils are, but I'm more interested in how we can remediate soils which have metal contamination so it doesn't become a public health issue. So
Hmm.
it's not an inhalation of metals by people, not ingestion or even leaching of the metals in the waters, groundwater, so people are getting exposed to that. So my idea for that research is using microbes to help plants grow in these toxic conditions, and as a result, prevent a public health crisis.
Yep.
Totally different, two different things, but very relevant to
It
where I
they're
am.
they're different but very similar as well.
Yeah,
Very similar, and there's a great need, especially for the metals one, with all the the bad things humans have done. The Superfund sites, in the
yep,
U.S., and the need for the mine tailings in areas and things like that. And now we have an even greater need for microchips and other rare earth metals and all this. So our need in society for these products isn't going away. So the
yeah.
need to be able to remediate soil will be even more important.
Yeah, that's true. We keep talking about, so there are other projects in my lab where we're trying to recover rare earth elements from waste that has been generated from different
Hmm.
mines. So if we can extract a valuable amount, then we don't have to mine more for those. So if there is a technology which can do a good job of, because we have to keep in mind that the older technology wasn't very efficient, and as a result of that, it has resulted in a lot of waste, toxic waste that is sitting on natural lands across the country. So with yeah, what you said is right. We want to be more green and we want electric cars, so we need more of those metals.
It's not one thing; it's another. Uh,
Yeah.
Hey, hey, hey! It seems never ending. But I have hope with
Yeah.
researchers like you. I have hope we can come with some solutions um, to make
Yeah,
the
my
world a
hope
better
is that
place.
too. Yeah, that we can contribute to help.
So, uh, what are your next plans? What's the your future looking like this coming year?
So this year, I'm actually not studying desert soil. I'm working on some soils from Poland, which are also heavily contaminated with metals. And what I'm trying to understand is we're looking at a plant called Arabidopsis halleri. And this plant is called a hyperaccumulator, which means is it has the capacity to uptake metals in its shoot and leaves. And there has been a lot of research done in this plant to study, well, how has it genetically evolved over time to uptake those metals, and that can have potential in phytoremediation to have plants that can be grown in metal contaminated areas and sort of lock that metal in a place and in the plant part, so that ways it's not leaching out and then it's keeping the soils from being blown away as dust. And
Mhm.
what I am adding to that project is looking at the microbial components and seeing if there are specific microbes in different soils that can be helping with this metal uptake. And what interesting fact about this particular plant is, so I'm looking at four different locations in Poland, and two of them have heavy metal contamination, and two of them don't. But there is a specific population of the same plant which is accumulating zinc where there's not enough zinc in the soil. And that just boggles my mind. How is that happening? If there's not enough zinc that we are measuring, then how are the plants accumulating similar levels? So we're hoping that the microbes are doing something to make
Hmm.
zinc available and the plants are uptaking it. So I'm going to try my best to answer that question.
That's nice. You have a
But
nice control site and a test site. That's convenient.
yeah, and so the PI I'm working with on, she has been studying these plants for a while, and that's why, and she moved from Poland to Tucson, so that's why we have access to these plants and data from that project. So yeah, I am adding the microbial part to it.
That sounds really cool. I always think about Arabidopsis thaliana, which is the more
Yeah.
popular uh, plant for biological studies.
Yep.
I believe it was the first one to have its genome sequenced.
Yep. That's correct. Yeah.
Ah, I remember these things. But I mean, that's it's promising because then you can crossbreed these plants, and maybe look for that best combination of uptake of these chemicals using the best combination of microbes.
Yeah, that's the plan. So there's another, I'm talking about all these projects which have nothing to do with me, but they're happening because of what we are finding in different projects. But we do have plans not only to use these plans for phytoremediation, but also seeing if there is a capability of extracting metals from the plants. So, like taking the biomass of the plant, and since they have uptaken metal in them, so is there value to extracting, let's say, if this Arabidopsis halori is taking up zinc, can we extract enough zinc that, again, we don't have to mine, or we have to mine less because of that? So it has another application to it as well, not just
Mhm.
keeping the metals locked in a place.
Yeah. So earlier, Michael was not asking, but wondering, and I love wonder statements. I use those in class all the time. I wonder, are there factors like water source chemicals that can harm microbes?
I think, yeah, it depends on, like, I think higher concentration. If the microbes are not specifically, I would think that they're harming them. And we have to keep in mind that microbes are more resilient in general because they're able to mutate
Hmm.
quickly given a scenario. But I think it's just the disbalance of nutrients or pH changes that could affect them, but I wouldn't say necessarily that they are like harmful overall for the microbes or bacteria.
You also mentioned if they're getting like if it's really dry, they might turn into spores,
Yeah.
and you just
Yeah. So
wait for it to rain again. And
yeah, so they have like mechanism. They have very strong adaptation mechanisms, so they're able to deal with a lot of different kind of. If there's more salt, they will adapt differently to that. They will use their osmosis regulation to control for that salt doesn't affect them. So they have different mechanisms to deal with it.
hmm. Dash, who just got here, asks, "What kind of microbes are present in crops?" Now, do you mean in crops, like inside them, Dash, or do you mean like in the soil? Because there are, I learned, microbes in the crops themselves.
Yeah, so there is a term called phylosphere, which means the microbes in the leaves of the plants. So there is a way of studying them too. But and then also you can look at microbes which are right at the surface of the roots of the plants, which are called rhizosphere. So which are not exactly inside because you don't have to cut open the roots to find them, but they're still like closely interacting with the roots of the plants. I am not 100% sure what kind of, because I don't study crops at all. But I would think that they will have different combinations of actinobacteria, protobacteria, which are the major acidobacteria. I think the major phylum, they should be present. And depending on if there are synthetic fertilizers added and what if they're nitrogen rich, then they might have different bacteria which can survive in those conditions.
So I did have, here we go. What Lorenzo Washington was on uh, Connected Conversations last year, and he does research on the fungi and the relationship with crops. So I will share out that video um,
Yeah,
if you're
that
interested.
yeah, that will be, yeah, more relevant to that question.
So he does plant microbe interaction as well, but dealing with
With
agriculture in
yeah,
particular. So
yeah.
there you go. You can go watch that later.
Okay. Thanks for sharing that.
Um, so we love talking about microbes around here because they're everywhere. You can't get away
Yeah,
from them.
yeah. Can I share another cool fact that a lot of people might not
Oh,
know? It's not
please.
related to the research that I'm doing, but
Desh loves facts. He loves
so
random science facts.
it's like a buzz thing in the agriculture because I after I finish my postdoc, which will be like next year, I would want to find a job in industry, and I'm looking at agriculture based industries where I can use the microbial knowledge that I have and help more sustainable agriculture. And what I'm finding is that these companies, like instead of the synthetic fertilizers, they are coming up with these microbiology, like
Hmm.
bacterial things, strains that can be used instead. Right now, not instead. They're not 100% replaceable, like replacing the chemical fertilizers, but in combination, so reducing the amount of chemicals in fertilizers, use half of this, the chemical fertilizers, and then the bacteria, because these bacteria are helping fix nitrogen from the atmosphere and providing it from in the form for the plants, which was essentially what the chemical fertilizers do. They provide nitrogen in a form that plants can use. So using the power of soil microbes to take out the chemical fertilizers, because there are hazards of using chemical fertilizers too. It can leach into the water, and most of the fertilizers are not uptaken by plants, so they remain in the soil. So that has effect on the wildlife, or wherever the runoff is happening in that water stream. So, in order to tackle that problem, we're trying, these agricultural companies, they're trying to use microbes, which I find very fascinating because I feel like this is something that I have been thinking about in terms of like the metal uptake part. If we can use specific microbes that we know will help uptake more metal, we can remedy it better. So I think there's a parallel to both these fields, and tackling one in the environment aspect and the other also sort of environmental, because if you have more leaching of these chemical synthesizers, then you're affecting the environment too. But in the agriculture setting, you're also producing food for people. So there are two things you're looking at when you are. So I find it very fascinating, and I hope that's something new that you all learned today.
Wow. I learn something new every day. This is great.
Awesome.
Um, so Michael, I like your comment. I wonder if there was like an indirect impact from the microbes we consume. You always wonder, like, all right, did I clean off my potato enough, and how much dirt am I eating as I'm eating my potato, right? And there, right, or like your other tuber, your root vegetables in particular, right? And it's like, I always just figure, it's probably good for you. Little dirt. My healthy bacteria in my gut will take care of everything, except for the ones that cause me disease. Uh,
I think for most part, yeah, like a little bit of won't hurt you, but accidentally there was a pathogen, because soils do tend to have some Pseudomonas aeruginosa and Staphylococcus. So those are not the ones that are good for humans. So if accidentally that is in the soil from where your vegetables are coming from, and they weren't washed properly, there's chance, but it's very, very little chance. It's not a high chance. Otherwise, we would hear more people getting sick.
mhm.
So I think very little chance of that
Yep.
indirect exposure.
Yep. Haxanet Tech. I watched a documentary on microbes and larger organisms in the soil, breaking down organic matter and moving nutrients from the surface down to the roots. What would be the best way to bring those into sterile soil for established indoor plants? That might be beyond our expertise here.
Yeah, I I think they might be referring to fantastic fungi. At least that's the one that I have seen, which is the most amazing documentary I've seen. It's just like it has been filmed very beautifully, where
I'm gonna have to
different kinds of.
watch that. Have I not seen that yet?
It's now on Netflix. It wasn't
Oh,
on, I
okay.
think it wasn't available on Netflix until recently. I watched it, I think, before the pandemic, so maybe like a year and a half ago, but it's pretty good. Yeah, sorry, I don't have a good answer to that question.
Fantastic fungi. Is that what
Yeah.
it's called? All right. My viewing is increasing. Also, my oldest child, she does the same thing that I do, in
Hmm.
that like she'll watch a lot of documentaries just because they're they're fun, especially like nature and like type documentaries. There's something
Yeah,
nice.
yeah, they're fun to watch, and also I always learn something that I don't know, so that's always fun to know more about what's happening in other species in the on the planet. It's always fun to learn.
You're a little hesitant to bring outdoor soil into the house. Hmm. What's the worst that can happen?
No, I don't, as long as you, well, okay, no, there is yes and no to that answer depending on where you are bringing that soil from. So, for example, if you're living one of the sites that I've worked the metal contaminated soils that I've worked here are from Superfund site. So if people who live around that community, they bring in outdoor soil and they are closer to that Superfund site, then that can be harmful for them. But if you know that there's no contamination that will affect you, then it's not such a huge issue.
So if like you just go out and get some soil out of your garden.
You are fine. I don't think it's going to be an issue.
Yeah.
Yeah, just wash your hands and don't try to eat it. Then you should be good.
It's someone trying to eat like a spoon and a bowl.
Spoonful full.
Mmm. Soil. Yummy. Gar, gar, gar. Okay. Thank you, Mrs. Jinzel. I've never tried that. I add some plant food once in a while to my indoor plants. That's about it. But maybe there are beneficial microbes that you bring in from outdoors. That helps.
Yeah, there could be a possibility of mixing some with your potting soil or whatever soil you're using indoors and doing that.
Hmm. Hmm. Yeah, well, that's why they say when you transfer plants outside, you try to get as much soil from around where that plant is growing to
Yeah.
then move it to someplace else. That requires a lot more digging, of course, but the more soil you get, the better.
Yeah, it's just because they're adapted and then they're soil microbes. So
Hmm.
everything is sort of set for them. And now you're trying to relocate. So you want to mimic or keep the environment as similar as possible so they're not in a shock when the plants are moved. And even like if you just planting from a little pot to a bigger, I have killed house plants because I wasn't careful. So yeah, they're very sensitive.
If a plant can survive me, it's a pretty hardy plant. All right, some good stuff going on in the chat. Now, I think that's kind of what we were talking about, Michael, where we're talking like those companies are trying to um like make these microbe, fertilizer things to sell us to
Yeah.
then, put into there, and there's, you know, it might help, but at the same time, you know,
Yeah, I do see advantage to that. And also, one thing that bothers me is also, which one of the things I mentioned is the microbes live in a community. So when you try to take them out of that, that can be challenging.
mhm,
So how they can perform solo is also something to look at.
right. It's kind of like I
Yeah.
think the probiotic movement in your yogurt.
Yeah.
It's it's like sure there's microbes in there, but will they provide you any benefit?
Yeah. And I think there's not
No.
conclusive evidence of that. Yeah. Yeah. Yeah, and
um,
also I think one thing we forget is one, the microbes live in a community, and also, sorry, what was I gonna say? I just lost the chain of thought. also how will they compete with new microbes. So
Hmm.
if we're introducing something in our yogurt which might be similar to something living in our gut, but it's not living in our gut currently, so there is some sort of adaptability to living in the gut versus being in the yogurt. So I think there will be some sort of competition as well. And if that's true for soil microbes too, if you introduce
Hmm.
something that was modified a little bit in the lab and then used in soils. I'm sure there's
Hmm.
more research, profound research being done, which I haven't followed completely, but it's a cool idea to pursue if it can help with less chemical fertilizers issues in agriculture
Hmm.
settings.
Well, I think we'll see more of it, especially with the popular or organic foods
Yeah,
and things like that. I think we'll see a lot more of this idea of healthy microbes and community into what you can buy at the your store for your plants.
yeah, yeah. I think, and I hope that because I love microbes. I feel like, oh, I hope it just works out. It's not just like something that created the buzz and now it's not like producing any results for people. I hope we are able to answer some problems with the use of microbes. That's my hope with everybody who's studying microbes.
Hmm. Is compost fertilizer richer in microbes, I guess, than industrial fertilizer? So I guess that's a curious question. How does your compost differ from other soils?
So compost will have similar. It will be comparable to soils like rich organic matter
Hmm.
soils, but in terms of industrial chemical I keep messing this up. Chemical fertilizers, it's going to be different because when we're talking about fertilizers, we are talking about three component nitrogen, phosphorus, and potassium. So it's a mix of
Hmm.
all these three nutrients. But when we talk about compost, it has more than that because it has organic matter, so it will have some carbon content too, so which is not necessarily provided by the chemical fertilizers. We in chemical fertilizers, we're trying to increase the amount of nitrogen that the plants can get. So there is a slight difference in terms of that, and that will also affect the microbes that are present because the composition is going to be different.
I'm curious, how many people in chat have a compost bin or compost at home? I'm curious. I do. And that thing is always just it never fills, but I'm always adding to it. It's amazing. You know?
I don't. I have thought about it, but then I think about the temperatures in Arizona. I'm like, it's too dry. It's not gonna
Hmm.
work. I probably have to do add vermiculite, so add some earthworms into it. And I'm not yet sold to that idea, so I haven't tried it. But it's on my mind.
Because I will say that's like one of the single best ways to reduce the volume of trash that you have.
Yes,
Because
that's true.
you produce, at least I do. We make a lot of vegetables, and there's always, you know,
Yeah, there's a lot of
so much.
scrapings. Yeah, yeah.
And
I
those
I agree.
bacteria love that decomposing matter.
Yes, yes, they do.
And also in on
There's
ground compost. Yep. Oh, yep. The rotating bin. My neighbor has a rotating bin.
oh awesome.
Yep. Um, you do have to be careful also if you have raccoons and other wild animals around. So
Yeah.
first you ferment your fruits and vegetables before tossing. If you're fermenting them, you got to make something out of them. Come on, Mrs. Genzel. Mine are usually fermenting in the bottom of my fridge in the supposed crisper. I think I have some vinegar in there.
Ah,
Funny. Um, yeah, yeah, composting's great, and that's like the opposite of what you research, like the arid lack of
yeah. It's
nutrient. It's
not.
like the opposite end of the spectrum. Lots of stuff, nutrients, and nothing.
Nothing. Yeah, definitely on the extreme side. Yeah. And I did buy compost for one of the experiments that I had to set up, and I went to one of the facilities in Tucson, and they told me that they do have to use some activators
Hmm.
to have the compost generated on their facility, because it's just, and they have to water it to give that moisture.
Hmm.
Otherwise, composting won't happen.
It'll just what it do? It'll just dry out and then blow away.
I guess, or yeah, probably just blow away, maybe, but mostly dry out and not really break down in any form.
Hmm.
I think so. I'm not 100% sure.
There you go. Leaves and sticks. Yeah, my I compost some leaves. You got to do the brown green. You're supposed to do a
Yeah,
proper mix.
yeah.
So I'll rake leaves and I'll put a bunch in there, but I don't put all of it. I just brush all my leaves in the rest of my backyard, and it acts as like a mulch. So, and the sticks, I just have a big pile of sticks in my backyard. But yeah, they take forever. And then of course, you gotta get your you gotta poke it with a shovel or something to get air in there.
Yeah, mix it. So yeah, there's aeration. Yeah.
So, do you speaking of aeration, so do you find most of the microbes aerobic, or are
Yeah,
most anaerobic that you work with?
not in the soils that I work with, because they are not inundated or at like lower depths in the soil
Hmm.
profile, so they are mostly aerobic. When I was doing my PhD work, then soils that I collected, they were from inundated areas, so they had like, or I was looking at sawgrass, and the soils that I collected from where I collected, they had like standing water in them. So
Okay.
those were the ones that I found more anoxic organisms in those soils.
Okay. Well, I think we are going to finish up. I have to like draw a strict line tonight as far
Sure.
as when I have to end. So this is the we'll do our last call for questions. If anyone else, I know if there's anyone else in chat has questions about soil microbes, um, we'll do a last call. What do you want to know? Um, I'll ask one last question since there's none coming up. We'll give them a second to type. Uh,
Okay.
what is the main um, metal that you're trying to extract? Is there one in particular, or are you just looking at all different kinds of metals uh, for the microbes that you're looking into for that extraction?
So there are two different sites that I work with. So the one from Poland, we are interested in zinc, because zinc
Hmm.
and cadmium are the two that Arabidopsis halveri accumulates. And then the other, the superfine site that I work with, it is high in arsenic, lead, and zinc. So for that, we are looking at a native plant species where we are trying to see if that species accumulate these metals.
Hmm.
Actually, this species doesn't accumulate it in high levels, but it does help if it can grow to sort of control that soil erosion and reduce any form of dust emission from the area.
Oh, here we go. They're rolling in now. Our last set of questions. What are your favorite microbes and why?
So my favorite microbes are the actinobacteria, and I was on another podcast and I shared this information, and this has been shared on an NPR shortwave story. There are certain groups of actinobacteria that are responsible for that nice smell when it
Hmm.
rains, and that's because they produce a compound called geosmin. And when it rains, the spores get activated, and that's the smell that of geosmin that we smell when it rains. Otherwise, they are all dried up, so we don't smell it. So I find them very fascinating, and it's a nice feeling when it rains in the desert. So definitely look forward to the smell of geosmin. That's why I find them. And also they have a lot of adaptations for extreme environments. So I think they're pretty cool and resilient.
Okay. Um, Question about okay, going a little creepy here, a little dark. Um, so dead animals in soil and microbes. Um, so we don't do that. I don't know many people that bury dead animals to try to increase microbe level in the U.S. I think where you're at, it might be different. Um,
I have not actually heard about that, but I do know of
hmm.
using microbes for determining the time of death for individuals for forensic purposes. So I'm aware of that. Depending on how much the body has decomposed, there are some studies looking at that if they can tell that. So there's definitely change of microbes that will happen when there is a decomposing animal or human body in the soil or dead plants. Anything that can decompose, the microbes are gonna change over time.
And I guess I would be surprised because I am surprised 'cause I had never heard of that before. Like, I am not burying bodies in my garden. You don't mix dead things in food, usually. It's just not, I don't know. That's just not usually where I go. I don't learn something else tonight. I guess some people bury dead things. Your pet cemetery is your garden. But
Event Yeah, if you have a pet cemetery in your garden, then eventually it's gonna break down, get
yeah.
decomposed, and the microbes are doing it. Yeah,
Oh, yeah.
but purposely using them to grow plants, yeah, I haven't heard that. That's a new thought for me.
Yeah. Well, you learn something all the time.
Yep. There's some fun
Hacks
comments in
tech.
this.
That's more like I mean I mean you're kind of like tongue in cheek there, but you know, I'm kind of in favor of the full yes, I want to be a plant. You know, when my carbon goes elsewhere, my carbon finds a new place.
Yeah, in the plants.
Have it be a nice, beautiful flower. It'll probably be a thistle. Uh, all right, everybody. This was fun.
Yeah,
This
thank you,
was
Scott. This was lovely
this is
to answer
fantastic.
all these different questions.
And thank you, everyone in chat. You were great. Really insightful questions, and we learned a lot because you were here to help us ask these great questions. And
Definitely.
Priyanka, thank you for taking your time and being here and talking
Sure,
to
yeah,
us.
it was my pleasure. Thank you, Scott. Definitely.
Yep. And this will be this is on the VOD, but it'll also be on my YouTube channel later as well. So you can check out whatever you missed there. And it's really not too long. It's only an hour long. So
Yeah.
it's a great listen. And thank you again so much. And everyone else, I'll hope to see you later in the week when I'll be on again Friday night. Okay. So have a good night, everybody.
Yeah, have a good night. Thank you for joining in.